Hello and welcome, everyone. We'll give a few more minutes. We hope everyone's having a wonderful summer, late morning, afternoon. All right, I think we have enough people to get started. Well, welcome. Thank you for joining Inno and me on this wonderful afternoon today. For everyone unfamiliar, and if this is your first Inno event, BostInno is part of a growing network of sites. We're currently in 14 markets. We're part of American Inno. We're a media and events company, and we primarily reach our audience through our core product, which is our daily newsletter, The Beat, monthly events, which are now all virtual. Please check them out. And if you subscribe to The Beat, you get a full rundown of everything innovation related in your city. What brings us here today is BostInno's newest virtual event series called Bytes with The Beat. We've modeled it on our afternoon newsletter that goes out every day. Sign up again. The series is intended to have or be a fun, short, yet informational conversation with people, movers and shakers in our innovation ecosystem that we continuously cover, and people you might hear about in our daily reporting. To kick off our first and inaugural Bytes with the Beat, we have with us Ashley Gard, biomedical scientist at Draper. We're going to be talking about Draper's new research in partnership with UMass Medical School and how its new device, organ on a chip, will be impactful in COVID-19 research. Without further ado, I'll let Ashley introduce herself.
Hi, everyone, and thank you, Shri, and everyone else at BostInno for inviting me to be a guest here with you today. It's really exciting to be a part of this kickoff for this new virtual series. So thank you for having me. As Shri mentioned, I'm a biomedical engineer at Draper. I've been a member of the technical staff and involved in R&D research at Draper since about 2017. So I've been here for 3 years. I came from training in cardiovascular and pulmonary tissue engineering and regenerative medicine solutions. And so it was sort of a natural course for me to come into biotech R&D and to apply all of that knowledge and training I had to work specifically on something we call organ on a chip technologies or microphysiological systems. And so my time at Draper has been solely focused on that. I work on development of novel tissues in unique platforms that are at the cutting edge of technology to really advance drug discovery and development. And so we're working in partnership with both commercial and government researchers to develop models of both healthy and diseased tissue, and then utilizing those models that we've carefully characterized to screen for treatment solutions. So that might be drug candidates, antiviral therapies, antibodies, and more, things related to the vaccine pipeline and such. So that's an overview of who I am, but I'd love to speak a little bit more to who Draper is to better inform everyone who's tuning in. So Draper is an independent research and development company based in Cambridge, Mass. We're here in Technology Square, which is just down the street from Kendall Square. Some of you may have walked by us. We have a big giant moon hanging in our atrium, so we're sort of a distinctive space, and come by and say hi. At Draper, we design, develop, and deploy advanced technologies to address the world's most challenging and important problems. And part of the reason why Shri invited me here today is to talk a little bit about our research on one of those really important problems, which is the COVID-19 crisis, and how Draper is getting involved there. And we are involved in various fronts, but I'm going to talk to you a little bit more about one particular approach to addressing this crisis that I'm intimately involved with.
So I'm sure, you know, this is a question that I'm asking on behalf of a lot of us attending. Is organ on a chip is it as literal as it sounds? Walk us through what it is and its impact or its role in COVID-19 research.
Yeah, so it's more or less what it sounds. Some of the earliest versions of organ-on-a-chip technology really was something that you could distill down to a micro scale single tissue that set in plastic hardware that more kind of resembled a microchip. So it's a tiny, small miniaturized tissue. And there are many versions of these organ-on-a-chip technologies out there now. But Draper's unique system is called Predict 96, and that's what we're going to be talking with you about today. And this particular platform allows for an increased throughput. So growing something on the scale of 100 of these micro scale tissues on the footprint of a standard cell culture plate. So we'll be able to show you what that looks like shortly. We've been, as Shri had mentioned in the introduction, we've been in partnership with UMass Medical School for some time now. We've developed a lung model of the upper airway in this Predict 96 organ-on-a-chip device. We are capable of recapitulating lung structure and function in this platform, and had been utilizing this in collaboration with UMass Medical School to develop a model to study influenza infection of the upper airway, allowing us to really tease apart what's going on the cellular level in this complex tissue, maybe how different patient samples respond, so looking at individual different donors, donor tissues behave, and then of course to identify novel therapies and ways to combat influenza that go beyond things that you might commonly know out there like Tamiflu, which is one solution for treating influenza. And so having developed this platform for evaluation of other respiratory pathogens, influenza, it was a pretty natural step forward for us to move into evaluation of other respiratory pathogens, that being coronaviruses. And that's really how we got to where we are right now and had such immediate impact in this space. And we're working and chipping away at this at the very moment, as so many of you probably are who are tuning in.
So what is, I mean, you know, there has been so much talk about everything that's happening, just emergency use, emergency authorization, everything. I feel like the biotech industry is getting on it now because this is their moment to reckon with. But can you tell us a little bit about why or how this can be instrumental in, you know, drug discovery and development, as it pertains to human trials? What is the impact that it could have?
So we feel that the Predict 96 organ-on-a-chip technologies really do provide a promise of a better future when it comes to identifying novel drug therapies. And that's because right now, about 1 out of 10 drugs that make it to human clinical trial eventually get FDA approval. And so, and then the cost of bringing that single drug to the market is on the order of $2.7 billion. That's a lot of money. It takes a lot of time. And you can see that you have to narrow down your candidates, and you still might miss the mark at the end of the day and have, you know, put a lot of money into this. So slow and expensive. What Predict 96 offers is an improvement in vitro systems that model human beings. And this can provide you insights into what therapies might be most efficacious and safe for a particular population of people. So you can think we can model both young and old, different sexes, cells from patients of different demographics on this platform. And so we can really begin to custom tailor solutions for different patient cohorts. And so the throughput of the platform and the fact that it creates this really relevant physiological environment and has this complexity that goes beyond what's considered the state of the art for drug screening in human cells right now, so that the standard is evaluating drugs in cell culture lines. So this is looking at single cell types derived from humans, maybe sometimes animals, can be derived from healthy or cancerous cells. And they're grown in a standard tissue culture plate or a petri dish. And these simple systems are highly reductionist, but they can be useful for just doing things in high throughput and moving quickly. But it comes at a cost because it's never going to accurately capture the complexity of a whole organism. And that's why we use preclinical animal models to glean some insight before moving to human clinical trials. And of course, with the COVID-19 crisis, there's been some jettisoning just quickly to human clinical trial because there's a need. But that, of course, comes with some associated risk, right? So we're trying to find a way to fill this gap in between those simple cell culture models that are just cells in a dish, and then these more complex organisms. And we fall in that in between with Predict 96. That's what we offer drug, pharmaceutical companies, government agencies, and partners of ours to glean some insight into what a human being's response would be at the tissue level or system level, so that you can knock out some of your drug candidates, hone in on the ones that seem to be most efficacious and safe, tease out what dosing might be most relevant before proceeding forward with these expensive preclinical trials. Use of preclinical animal models is expensive, and sometimes these animal models don't exist for specific conditions. There's just not a suitable animal model out there. In addition to the fact that they're very expensive. If you wanted to utilize non-human primates, it's actually quite hard to get those at this time with regard to the COVID-19 crisis. So you can see there's a pretty significant need for something that allows us to gain information about a human's response to these potential therapies. So Draper's really hoping to change the timeline and the cost associated with bringing a drug to market, and that's what Predict 96 is gonna provide companies and individuals out there looking to do this type of work.
Yeah, and I mean, there's something you said that I want to kind of go back to. As a scientist, there are processes that you follow, especially in a lab, before a drug goes to even a trial or discovery. But how did all of that change with the pandemic, right? Like when you realize that you have the power to actually cause action and do something impactful as a scientist, and as a scientist working on COVID-19, what were some of those complications?
Sorry, just to repeat. So the complications with moving forward?
With moving forward and also working with this technology as it pertains to COVID-19.
So at this time, we've been doing a lot of our initial work with what is considered BSL-2 safe coronaviruses. And one of the complications is just getting access to spaces to do this research and to do it safely. There was a lot of concern initially that it was challenging to work with SARS-CoV-2, which is the virus that causes COVID-19, due to the associated risks. Since starting this work, though, we have now secured partnerships in our own BSL-3 space, so we'll be able to move into handling SARS-CoV-2 ourselves. But up until now, the immediate solution was making use of some of these common cold-causing coronaviruses to more or less validate that our model can support being infected with this particular pathogen, that it can enter the cells and it can replicate, and we can model that. And then, of course, to screen with some known therapies that might block or prevent replication of those coronaviruses. We evaluated those to sort of say, yes, our system is capable of not only recreating a coronavirus infection, but we can actually prevent that or reduce that coronavirus infection. So the natural step forward for us right now, and what we're working towards at the moment, is just moving into that BSL-3 facility and working directly with SARS-CoV-2 so that we can gain those insights and begin screening some of these potential therapies in partnership with clients of ours or collaborators. I'm not sure if I answered your question exactly, so feel free to ask.
You did. And that is exactly what I was looking to ask you. I think the question sort of came up in my mind because we're learning so much about the disease every day. And how does that, how do scientists or the scientific community use that information in their daily research, right? So that's mostly where my curiosity was coming from. But maybe you can talk about that too.
Yeah, I mean, this has been an evolving process, and a lot of the data that's emerging is being, presented online on open source sites such as bioarchives and medRxives. And so our engagement with this COVID response community in Greater Boston has been really important for facilitating conversations between research groups and academics and clinicians. And so it's really about keeping on top of what's emerging from the literature, going to credible sources and extracting that information and trying to distill it. And that definitely has shaped our thinking and our hypotheses in the lab and our strategies. We've been grateful, fortunate enough to identify some really great collaborators, and we're looking forward to working with those teams. I think that it's going to take the entire scientific community really rallying and coming together. We all come with different expertise and tools and technology to offer. And so here at Draper, even though we have, excellent biologists and engineers that sit side by side, allowing us to do this iterative process of design hardware, evaluate the biology, look at the biology, have that inform hardware design. We absolutely turn to the experts in other respective fields. World-class infectious disease researchers like those at UMass that we partner with, but many others, virologists, immunobiologists, all these people have been important to us in shaping our understanding of this disease. And it is an ever-evolving process, and we're excited and eager to assist here. And I think that PREDICT can help us glean some insights into novel aspects of the disease pathophysiology, because we can have that window into the tissue that you can't get when you're working with a whole organism. You may have to, unfortunately, sacrifice that organism and terminate its life in order to get some snapshot in time. But the PREDICT platform uniquely allows us to take a look at the tissue at any point during its growth. Maybe that's before infection, maybe that's during infection. And so we're hoping to contribute to this community by getting that window in and really helping people see what's going on a cellular level. How are these cells interacting in a tissue? What immune cells are they interacting with, and how this disease plays out. And I think that's kind of the crux of it right now. We need to understand what's going on a mechanistic level to be able to identify the right types of therapies. And so we're very excited to work with future clients and customers and collaborators to make this possible. We look forward to all of those future engagements. At some point, I would love to queue up a video.
Yeah, we have buried the lead enough. I guess it's now time we learn how it works.
Yes. So we'll show a video that you can also find online later. It's a Vimeo video, and I know some of this content will be posted later. So here we go. So here is an example of the PREDICT 96 system. That lid that was just placed onto the plate allows for fluid flow. It recirculates in each individual device, and there's actually 96 of them depicted there. This device can integrate seamlessly with standard industry workflow and industry standard microscopy, fluid handling robots. And you can see here with this cutaway, you can get a window into the inner workings and the hardware. We have many micro pumps that control the fluid flow for each individual device, and they're pneumatically actuated, which means it's an application of air and pressurized air and vacuum that help drive the recirculation of the fluid in each individual compartment. Each individual tissue unit has actually 2 chambers, and what that gives you is an ability to introduce cells on both a top and the bottom of a semi-permeable membrane. So you can create a tissue barrier and model things like the lung. And here you can see independent fluid flow on both the top and the bottom of this human tissue. And so you can have multiple cell types introduced. You can introduce drugs on either in either chamber. And expose that tissue. You can, of course, in the context of COVID-19, introduce pathogen on one side of the tissue, on whatever side is most relevant for what you're looking at. We're capable of modeling a variety of human diseases as well as infectious diseases. I'm glad you got a little snippet into it. And please feel free to watch that video again. We'll make sure that it's accessible to you, but I do want to show you with it in hand what it looks like right here. You can see the device is quite compact. I can hold it with one hand. And what this means is it's very user-friendly, especially for someone working in a BSL-3 or a BSL-4 facility. It's really important because you're in additional protective gear. But even within a small laboratory space, you can grow and culture tissues in a number of these devices at once. You can actually get somewhere on the order of 16 to 20 something of these plates in an individual incubator, sorry, that you can see here behind me. One of those individual units, you can fit a number of these in, and you end up getting somewhere on the order of 1,000 to 2,000 individual human tissues growing at any given time. That is sort of what you can achieve at any given time in one of those incubators. So you can see the power of what that throughput affords you, and these dynamic systems that let you grow tissues that would otherwise be impossible to grow in standard conditions. So here I'm going to take off the lid, which is the pump. It has 192 small micro pumps that allow you to recirculate fluid flow on the top and bottom chambers of the device. The plate, as I mentioned, is a standard industry form factor. It looks just like a 384 well plate for those of you who are used to this sort of stuff. And I'm going to bring it close to the camera here, and you can see a little colored resin. And so there's a yellow on one side and I think a green or a blue on the other, and that lets you see the two chambers for one individual tissue unit. So there's 96 here. And we put the lid right back on. We would attach a little bit of tubing, and then it would go right into the incubator. So compact and easy to use, and lets you generate a lot of data at one time.
That's fascinating. So how do you, I mean, like, how does this replicate a standard human trial? Like you said, you know, there could be different interacting complications, right, when you actually do a human trial. But how do you kind of effectively translate that or try to get the data which would be correlated?
Yeah, I think it's really important to turn to human clinical data that exists or that's currently being generated. You need to look for metrics that are associated with whatever you're trying to study. So if you're, looking at evaluating COVID-19 therapeutic efficacy in a patient, you might look at functional metrics and readouts that would let you know that the therapy was in fact working or not. And so we then use those metrics. It might be a unique biomarker that's being secreted or not. And we then search for those same things in our platform. Now, a lot of this clinical data is being generated right now. So ideally, it would be run side by side, a human clinical trial to sort of say, yes, we've validated and verified that we are getting similar information in this platform. Now let's look at therapies that are also being used right now in human clinical trial. And I think that once you have the alignment there, you can be confident that the technology can in fact model the disease. And so that's sort of the gold standard of how we approach this and what we work towards. So I think, it's just going to be, like I said, a constant conversation with those that are involved in the treatment of COVID-19 patients to make sure that we're accurately capturing all features of this disease so that we can get a handle on whether or not, or a handle on which therapies are most effective.
And assuming that, we will one day put this pandemic and coronavirus, COVID-19 behind us, how does this, kind of, what role does this play in other types of drug development and human trials? It's other than respiratory diseases. What are the other, research that is going on that's involved with this technology?
Yeah, so we've been working on this technology for over 5 years now, and have had numerous customers come to us from the commercial sector and the government. And we've co-developed tissue models with some of these partners, groups like Pfizer and Bristol Myers Squibb and Colgate-Palmolive. We've developed different individual tissue models that were all intended with different purposes. So in the case of the oral gingival model that we developed with Colgate, it was to screen oral care products. Whereas with the other groups, it's developing a healthy and then a disease model for a specific disease and condition. And these, you can see here that, you know, it doesn't, this platform isn't just effective for studying COVID-19 or a viral pathogen, an infectious disease, although it has a lot of impact, it can have a lot of impact there. But we can study anything about the human tissue. We can look at bacterial pathogens, but like I said, we can study any sort of disease or inflammatory condition, and we could look at comorbidities. And there's also the possibility to link and have different tissues interact with one another. And so you can begin to understand how some of these tissues might interplay, and how maybe a condition in one leads to a different downstream problem in another. And I think you know, moving forward for us and sort of what's really next with this technology, there's a lot that's next, honestly. But in the context of COVID-19, you know, and I think this applies beyond it, we're constantly looking at sort of addressing depth. You know, how can we screen as much as possible, like convalescent plasma, antibodies, small molecule inhibitors rapidly, so that, you know, and a variety of therapeutic strategies, so that we can get to a solution for COVID-19 or whatever your disease condition is as fast as possible. It's going deep because we have the ability to. We have the complexity of the biology and we have the throughput. But then we want to understand breadth of conditions. So in the context of COVID-19, we might want to not just look at the upper airway, but we might want to look at the entire airway tract, down in the alveolar sac at the lowest portion of your lung where you're having gas exchange occur, all the way up into the nasal epithelium. And then we might want to go and screen, you know, vaccine products, do vaccine development work, screen antiviral therapies in those other tissue models. And but, you know, in addition to that, as I mentioned earlier, we may also want to look at different patient cohorts, young and old samples, different sexes, different demographic populations. I don't think there's going to be necessarily a one size fits all. We're all probably aware of the fact that COVID-19 is a complex infection and disease, and it's very possible that different demographic populations and different cohorts have to be treated very uniquely. And so we're hoping to provide that breadth with our platform and work with drug companies and pharmaceutical entities to really move that forward and learn what we can about that and find solutions that are unique to those groups. And then, of course, there's the complexity that can be achieved in our platform. We could have one given tissue type grown from, we'll say, 2 different cell types, but there's nothing stopping us, and we've done this before, and adding in additional cell types, adding in an immune component, a unique immune cell population. Draper's uniquely equipped to do this. We love innovating new technology. We sit at the fringe of science here. We love to create something that has never been conceived of before and push the bounds of what's possible with our technology. And so, as I mentioned, the depth, breadth, and complexity are all important points, I think, for driving home finding unique solutions for COVID-19. We know it affects lots of different tissues in the body, even if it is defined as a respiratory infection initially. But these things across the board, you know, these 3 things definitely apply across the board. And I think there's a lot of potential for this platform to grow and develop with unique questions in mind.
And yeah, and I think you've already explained the reach and the potential of it, and we're very excited to see where this goes. Hopefully we can all, you know, have an answer for what cures COVID-19 sooner. But I think that brings us to the end of our first Bytes to the Beat. Thank you so much, Ashley, for joining us today. I hope you had fun. There was so much to learn and there was so much to share. I'll leave you if you want to say a few final words about this.
Well, I definitely want to reiterate my thanks, Shri, for having me today. It's a pleasure. And like I said, it's exciting to know that I got to help you kick off the series, Bytes with the Beat. Woo. I'd love to encourage anyone who is was, intrigued by what I had to say or wants to learn more, to please reach out to us. You can find more information at draper.com. That's DRAPER.com. We're located here in Cambridge, as I mentioned earlier, and we do a lot more than just work in the biomedical space. So for those of you who have interests that span other subdisciplines, I do want to note that aside from a lot of our business being backed by the government and government funding, we also have a large role in the commercial market, specifically centered around transportation, energy, and then of course biomedical solutions. And we do serve global customers in biomedical solutions. So if you, for the viewers out there, if there's interest, like I said, to learn more or to interact with us on a business level, please don't hesitate to reach out. All that information should be on Draper's website. And I know Shri will be posting some additional content that relates back to this. So we're happy to share some additional resources to make engagement possible.
Yeah.
But thank you so much for having me. It was exciting to have a chance to talk about the work.
Thank you. Likewise. Thank you, Ashley.
Thanks.